Hello and welcome everybody to my channel ominibioentrance, and today we will be seeing CSIR PYQs June 2023 Shift 2. I will be dealing with applied biology and methodology, DNA replication, protein synthesis and genetics. These are solved questions of part C. If you are new to my channel, please do like, share and subscribe. I have also uploaded old CSIR PYQs. You can refer to my channel for the same. Coming to the next question. Given below are some statements with blank spaces indicated by a dash. So you have to do the fill in the blanks over here. The plasmid cloning vector digested with an enzyme with a single restriction site in the plasmid that generate 3' overhangs can be made blunt-ended using which enzyme? So you have a plasmid cloning vector. It is digested with an enzyme, and that enzyme, what it does is it creates 3' overhangs. So you don't want any 3' overhangs. You have to remove the 3' overhangs and make it blunt ended. Now that can be done with mung bean nuclease. So mung bean nuclease can be used for removal of both 3' and 5' overhangs from single stranded DNA to create blunt ends. So third one is the answer. A, mung bean nuclease. The other options, A, PFU polymerase, tag polymerase, reverse transcriptase cannot do that. Only mung bean nuclease can do. So your answer is option 3. We will see the others also. DNA with a nucleotide composition of 30% A, 35% G, 20% C and 15% T is more likely to be. So you want to know whether it is single stranded or double stranded. So if you see A is not equal to T, G is not equal to C. So it doesn't follow the Chargaff's rule. So it is single stranded DNA. So answer is B. B is single stranded DNA. Then next one. Production of only truncated molecules of transgene derived mRNA in transgenic plants generated using transgene from a prokaryotic source is most likely due to. So you have got a transgenic plant. It is having the transgene derived from a prokaryotic source, and it is producing truncated molecules of transgene derived mRNA. So they have asked why it is producing these truncated molecules. You all know that prokaryotes lack polyadenylation signal. So when a prokaryotic transgene is introduced into a eukaryotic plant, it will not contain the signal for polyadenylation and processing of mRNA. As a result, the transgene derived mRNA produced in the transgenic plant may lack a polyadenylation tail, and this lead to the production of truncated mRNA molecules which are unstable and might be rapidly degraded within the cell. So the answer here in C for C is presence of potential polyadenylation signal in the transgene sequence. Then D, dash is a method for identifying the positions where individual DNA binding proteins attach to a genome. So you have to see for chip sequence. Option D, that is chip sequence. So chip sequence stands for chromatin immunoprecipitation followed by sequencing. It is a technique used to study protein-DNA interactions in cells and also for identifying positions where individual DNA binding proteins attach to the genome. So the answer here is option 3. The following represent selected AFLP bands, that is 1 to 5 observed in parents P1 and P2. F1 progeny and 20 double haploid progeny developed from the F1. Double haploids are created through chromosome doubling of pollen grains in anther culture. The following statements were made about the above AFLP bands. Bands 1 and 4 are allelic. If they are allelic, wherever bands for 1 occur, there will be no bands for 4. And vice versa. So bands do not occur here but in 4, but bands occur in 1. So here no bands are there. Here bands are there but no bands in 1. So they are allelic. So statement A is correct. Now bands 2 and 5 assort independently. 2, 5. Now if there must be 50% parental and 50% recombinants. Only then they will assort independently. Now we will see which are recombinants and which are parental. Now if bands are present both in 2 and 5, they are recombinants. So 1, 2, 3, 4 and 5. 5 recombinants. Here bands are not present. So these bands are parental bands. Here these are parental bands as bands are not present here. So how many parentals? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 parentals, 5 recombinants. No, there are other recombinants also. If bands are not present in 1 and 5, then they are recombinants. So how many recombinants altogether? 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So 10 recombinants, 10 parental. That is 50% parental, 50% recombinants. So you get they assort independently. Band 3 is uninformative. Yes, you get this band, it is uninformative. So C is also correct. Which of the following option represent a combination of all correct statements? So A, B and C are correct. So answer is option 4. Coming to the next question. The following statements were made about X chromosome inactivation in humans. Maternally derived X chromosome has a greater chance of becoming inactivated in any given cell. That is not a correct statement because in X chromosome inactivation can be either to the maternal or the paternal chromosome. It can be either of them, not that specifically maternally derived X chromosome is getting inactivated. So A is not correct. Both X chromosomes are activated during the process of oogenesis. That is correct. The XIST gene encodes for single long non-coding transcript which bind to the X chromosome and helps in its inactivation. That is correct. That's the function of XIST. The XIST gene expression is required to initiate inactivation of X chromosome and also to maintain inactivation from one cell generation to next. So the XIST gene expression is required to initiate inactivation of X chromosome is correct. But for maintenance of this inactivation from one cell generation to the next is not correct. So option D is not correct. TSIX transcription affects the abundance of XIST RNA in cis. TSIX is transcribed antisense to XIST, so it regulates XIST RNA expression. So the E is also correct. So B, C, E are correct. So answer is option 2. Then next one. In an experiment, immunophenotyping of human blood cells was done to analyze the relative abundance of CD8 plus T cells and CD4 plus T cells. The following antibody fluorochrome conjugates for staining different blood cells were available. So for B lymphocytes, CD19 FITC was available. For T lymphocytes, that is CD8 plus cells, CY7PE was available. For T lymphocytes, CD4 plus CY7PE was available. For CD3, for T lymphocytes, both T lymphocytes, CD8 and CD4 plus CD3PE was available and DAPI for nucleus. Which of the following option gives the correct sequence of the antibody fluorochrome conjugates to be used to sort the relative abundance of CD8 plus and CD4 plus cells in the given blood sample by FACS? So here you have to find from these different stains what is the sequence of the stain that you will use so that you can sort out CD8 plus and CD4 plus cells. So the answer here is option 4 because first is DAPI. Now DAPI, it is used for staining the nucleus. and it is used to identify cells based on their DNA content. It also helps to distinguish between live cells and dead cells and can be used to select the population of interest. So first you have to stain the cells. You have isolated the cells, then you have to stain them with the DAPI nucleus. So you will come to know which all are live cells and which are dead cells. From that, isolate the live cells and keep it aside. Next, you have to stain with CD19 FITC, that is for B lymphocytes, and CD3PE for T lymphocytes. So CD19 FITC and CD3PE, of these CD19 is a marker for B lymphocytes and CD3 is a marker for T lymphocytes. So FITC and PE are different fluorochromes to label these antibodies respectively. And CD19 FITC and CD3PE are used to identify and B lymphocyte and T lymphocytes, and from these population you can exclude the B lymphocyte population, because you need only T lymphocyte population for analysis and not B lymphocyte. So first you have stained the nucleus, found out the live and differentiated between the live and the dead cells. Then you have isolated the live cells and chosen for T lymphocytes alone, excluding the B lymphocytes. Then next was CD8CY7PE. So CD8CY7PE, I told you, used to CD and CD4CY7PE. These are different subsets of T lymphocytes and used to label different subsets of T lymphocytes. Okay, these two are used to label different subsets of T lymphocytes. CD8CY7PE and CD4CY7PE are used to distinguish and identify CD8 plus T cells and CD4 T cells respectively. So first DAPI stain, then CD19 FITC and CD3 to distinguish between B lymphocyte and T lymphocyte. Then CD8CY7PE and CD4CY7PE for distinguishing between and sorting out CD8 plus and CD4 plus cells. So this is how you do immunophenotyping of these blood cells. So that is the answer. Then next one. Given below are few statements on mapping population and marker assisted selection. Which of the following option represent a combination of all correct statements? So marker assisted selection can be used to eliminate undesirable genotypes early in the breeding program by screening plants at the seedling stage. That's correct. In backcross breeding programs, breeders use molecular markers to select against the donor genome to accelerate recovery of the recurrent parent genome. That is also correct. Among different types of mapping population, F2 and F2:3 population are immortal. F2:3 is not immortal. So C is wrong. Near isogenic lines can be produced by repeated self-pollination of F1. They can be developed by selfing or backcrossing of F1 with recurrent parent. So D is also not correct. So the answer here is A and B, that is option 4 is the answer. Two mutations were isolated in bacteriophage, one causing clear plaques and the other minute plaques M. The genes responsible for these two mutations are 9 centimorgan apart. The plaques with genotype C plus M minus and C minus M plus, these are the parentals, were mixed to infect bacterial cells. The progeny plaques were collected, cultured and plated on bacteria. The expected number of different types of plaques are shown below. Which of the following option represent the combination of all correct statements? So your answer is C and D, and I will tell you how it is. So these are the parental combinations. C plus M minus, C minus M plus, C plus M plus, C minus, M minus, they are your recombinants. They are 9 centimorgan apart. So 9%, so 4.5% each. So the remaining will be 91%. That will be your parental genotypes. So 45.5 and 45.5% each. So C is correct. C, if you see C plus M plus, that is your recombinant, is 45. C plus M minus is 455. That is your parental. C minus M plus, again parental, it is 455, and C minus M minus is 45. That's your recombinant. So C is correct. That directly shows the answer. C is correct. Now D. Now D, if you add all these 65, 680, 685, 70, you get total of 1500. Then you see for the distance. 65 by 1500 into 100 will be 4.3. 70, that is your other recombinant. 70 by 1500 into 100 will be 4.6. These two values are approximately close to 4.5%. Now 680 by 1500 into 100 will be 45.3, and 685 by 1500 into 100 will be 45.6. So these two values are also close to your parental genotype. So D is also the answer. So the answer here is C and D. Then a mutant DNA polymerase was found to have high error rate and synthesized only short DNA fragments. In the statements below, potential explanations are given. The 5' to 3' exonuclease activity is compromised. No, that is not the correct answer, because it should be 3' to 5' exonuclease activity, that is proofreading activity, that is compromised. So B is correct. The polymerase tend to frequently dissociate from the template. That may also be the reason why you get only shorter DNA fragments. So C is also correct. The polymerase is unable to unwind the DNA. They have not mentioned that the mutation causes unable to unwind the DNA or anything like that. So that is not the correct statement. So B and C is the answer. So thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, do subscribe. I have also uploaded questions from previous year CSIR exams. You can go through that in my channel. Thank you.